all, I would like to thank the organizers for having me here. Special thanks to Daniel and Juliet. And this is like very nice event to share knowledge with other experts in the field. So I'm Ismail. I'm going to present SuperNet, Super Resolution Network Analysis and Quantification of Single Molecules. This work has been done with Prof. Hamarni from Simon Fraser University and Prof. Nabi from University of British Columbia, both labs in Vancouver. The theme of my talk here is like to leverage machine learning and network analysis for macromolecular structure determination from SMLM. So I will focus here in the biological application, which is related to cabulin-1 protein, the clusters of cabulin-1 protein that we are studying in the cell membrane that forming some macromolecular structures, such as cavuli. These are invaginations in the cell membrane with size of 80 to 100 nanometers. So it's like below the diffraction limit. So super resolution is good image modality to study and understand these structures and their features. As well, we are studying scaffolds, these another kind of clusters of Cav1 proteins. These structures still like unknown structures. Right? So the cabuli have been studied by EM microscopy. So the motivation here is to use super resolution and then apply computational methods and algorithms to detect the clusters and identify their features, and then relate this to the biology and answer the biological questions. We face challenges here, as all of the people who work in super resolution, as I get this from the talks. So it's big data. So we have large field of view in our in-home microscopic data set. So we have almost 54 micron, and we have millions of localizations. So it's big data. And we are not interested in analyzing small regions of interest. We are interested to analyze the whole field of view for the data. It's a noisy a little bit. So due to ground and different sources of like artifacts, multiple blinking of single fluorophore, etc. And not much work in the automation of such a data and the quantification for 3D super resolution data. And I would like to say about like the data-driven approaches here. It requires ground truth. So the ground truth here is like limited, and we are working in like sometimes in biological discovery, so we don't have ground truth, and no one can provide such a ground truth data. So let me show you the pipeline that we proposed to analyze super resolution data. So here we start with a super resolution image, which is 2D, acquired using GSD or DStorm, Palm, etc. We are working in the corresponding 3D point cloud data, and we can apply an optional step here, the merging, to correct for multiple blinking. So our goal here is to make it artifact free. We made it like optional. I will show in the implementation for this pipeline. And we can work for the whole field of view, or we can divide the cell into regions of interest. And now we start by constructing the network. So the network, it's a graph. The graph is a set of nodes and edges. The graph, it's like rich data structures that encode the relation between the nodes. The nodes here are the molecules in our case, or the localizations for the molecules. The edges like incorporating the interaction between every pair of nodes in the data. So we construct a graph, regardless if it is like 2D or 3D. Now we have planar graph. And once we have the graph, we can extract per node features. So this is like the local features, and we work at the local scale, or we can extract like mesoscale, which is like cluster or subcluster features, or at the graph level features, which is like global features. So we can, based on graph theory, to extract so many features from this enriched data structure. And after getting the graph, we can label the nodes according to the features. For example, here based on node degree, or even we can have weighted edges here. The weight here is like the strength between like the interaction between two edges. So we can label the nodes, and we can reason about these to do different tasks. For example, here, if we want to filter out the noise, so we can compare the features of the graph with random graphs. So we can retain the nodes that have features different than the random graphs. And at the end, we have the clusters. And this is like the denoising step. The next step is to delineate the individual clusters. We call them blobs here in our work. So these are the clusters that we're looking for. So we delineate these by the segmenting the blobs. And once we have the clusters, we can extract features for every one of these clusters or blobs. The features that we use comprise shape features, network features, statistical, topological features, etc. So we extracted 28 features for every one of these clusters. On top of that, in another work, we extracted graphlets features. Graphlets are like the basis of the graph, or like subgraphs or the patterns. And in this work, like we counted the frequencies of these patterns, and we find like, okay, every class or every biological structure have like frequencies different than the others. And this helps us to identify the structures. And finally, we use the features, either the classical features or the graphlets features, to identify the biological class for the structures. So this is the pipeline. And we used our pipeline, for example, to study, as I mentioned, the CAV1 domains. We identified four classes. So we identified 3 scaffolding domain and the cavuli, which is known here, this structure. And using our pipeline, we described the formation of cavuli, for example. So here we can see like the formation of cavuli, it's via 2-step process. So the smallest structures, we call them scaffolds S1A, that dimerize to form S1B, and in its turn, like dimerize to form the large structures, the cavuli. So the cavuli here is like one cluster, but we studied the subclusters or the modules or communities in graphs, the communities of molecules here interacting with each other. And we studied these and relate them to the other structures. And we are the first who studied the hemispherical scaffolds, described their features, and it's like first time we see such a scaffold. So here I'm gonna show like our results for the cavuli confirms with the results and extend other approaches, like some other methods used electron microscopy to study the cavuli, and they reported that the cavuli consist of 8S complexes, which is in our paper, it's like correspond to S1A scaffolds. So here is SuperNet software. So it comes to implement the pipeline. So SuperNet, it's like a graphical user interface, cross-platform, can run in Linux, Windows, or Mac OS. It's easy and user-friendly. It's based on graphs and network analysis, designed for 3D data. And it supports like a lot of features for pre-processing and post-processing, and a lot of visualization for the features and for the data. Our software equipped with machine learning for cluster identification and feature extraction modules. And it's a direct implementation for 3D SMLM network analysis, which was published in Scientific Report 2018. And SuperNet under development right now and in preparation, and we will make it like available for other users in different applications and different labs. Let me walk you through the SuperNet software, show you some features of the software. So this is the main window for the software. You can see here we have tabs. Every tab, it's like one stage of our pipeline. And here, for example, the load module. So you can use this tab to start loading your data. And you can see here we have channel 1, channel 2. This is for dual labeling or multicolor labeling data. But right now we're supporting just like channel 1, which is like just single label. And we are aiming for, in the next version, to have multi-label analysis. So loading the data enables the users to load the file. We support different file formats from several microscopies. So here we can integrate more formats. the user can process any data set in point cloud format. And once the data is loaded, it's gonna show like the number of localizations and the number of descriptors. So the descriptors here varies from microscope to another. And once the data is loaded, the user can quickly visualize some statistical features about the data. For example, the photon count, the X, Y, Z distribution, etc. And once the data is loaded, again, the user can visualize it. And here is the preview window where the user can change the color or code for the molecules based on different descriptors, and can use the color information here and color map. And here the interesting thing, the user can find some artifacts. For example, the photon count, he wants to neglect some values or some artifacts in the data. So he can or she can like move the sliders here. So he can crop region of interest, or he can filter out some artifacts from the data from the very beginning. For example, here we load the data. This is the Khater embed data. And we discovered that the Z here have a spike. And this is an artifact in localization algorithm used to acquire the data. So just moving the slider, we can get rid of this spike, and we can see the artifact-free data here.
Okay.
So we continue here with like the merging. It's an optional step here to correct for multiple linking. Once the user set this as a 0, and this means like merge is not applied here. And to construct a network, we here need to set the proximity threshold parameter and calculate the network. Then it's gonna show the data color coded by the network degree here, for example, one of the network measures. And we use the network features to filter out the cell. Here, for example, we slide the alpha. And here, let me show you the degree distribution for the network, for the nodes, and the degree distribution for the random network. And you can move the slider so you can filter out the noise. And we give the user the flexibility to filter out the noise based on this graph. So this is the optimal value for filtering. And here to show you the data before and after filtering. Then we need, as I mentioned, to delineate and segment the clusters into single clusters. We use mean shift algorithm to segment the blobs or the clusters here. And this is the segmented blobs. Then we extract the features, the shape features, the topological features, the network features, etc. And we can visualize them. Once we have the features, we can use the features to identify the groups. So here we identified 4 groups. So we color code the blob space and these groups. And we want to study the features for each one of the groups, so we can visualize the single or individual features for the blobs. Moreover, we can here use like a pairwise feature visualization to allow us like to select one feature in x-axis versus another feature from the same feature set to visualize them here. For example, we have 4 groups. It shows, for example, one of the graph measures or one of the graph features called characteristic path. It shows for the green group, it have like high characteristic path and low average degree, while the blue group, for example, have low characteristic path and high average degree. So the most interesting one for biologists here, this is by working closely with biologists, and they like this feature, like listing all the blobs as like the rows here. So each row here is like the blob color coded based on the groups that we obtained in the previous step. And it shows the feature of this blob. And the user can click at any one of the rows, and it's gonna show the volume, for example, for the localization of that cluster. So he can interactively see some representative blobs or clusters from his data. And at the same time, he can see the network between the, or among the nodes in the network, and he can use this slider to change the connectivity within the same cluster. For example, here at 40 nanometer, he can increase this and see how the network look like. So he can see some interesting modules in the same cluster, for example, or subnetworks. The same thing here. The interesting thing here, he can explore the blobs. And for example, he can press in one of the features here to sort them and filter them. And for example, if he's looking for the most spherical blob or the most elongated blob, he can just like click in one of the rows here, in the one of the columns here, and see all these features interactively. Okay, I want to mention that we use our pipeline in many publications for biological discoveries. And recently we published a review paper that studied like the state-of-the-art methods, and we compared the pros and cons of different methods. Let me like summarize the proposed method features. So the proposed method here, it's very fast. For example, like processing 1 million localization can take less than 1 minute in any PC. No need for server or cluster computing here. It's scalable, so it can be run on like cluster computer and can be used to process huge data sets for like many data sets at the same time. It's 3D from start to finish. Some methods like start like acquiring 3D data and use 2D methods. It's robust to noise, as we saw. Like we can process like data with like different noise levels, and we can get like the clusters. And it's flexible and general. It's not limited to the CAV1 domains. Can be applied to any other data sets, like Clathrin data or other biological structures. And the good news about this, like it's quantitative like features. We have features for the blobs that can be used with machine learning to identify the biological classes for the clusters. And finally, I would like to thank our collaborators, Prof. Hamarni, and my supervisor, and my collaborator, Dr. Nabi, and my collaborator, Dr. Chuz. And I would like to thank the